So if this material is interesting to you, and you might consider an intern, taking on an intern to help you further in this area, get in touch with me. I'm also looking for a postdoc to work on the brain coding that I'm going to talk about. It was 1989 that I decided that the area I was working in biology, viral genetics, was not going to be a rich source of income, an easy grant, and an easy way to go. So I came to UC Irvine and did a sabbatical with Carl Kottman to learn neurobiology. And coming back, I was in between grants funding, and I thought, well, there is a need for a better neuron culture model, to be able to do neurotoxicology, because after all, a lot of the bad things around us affect our brain, neuropharmacology, to put drugs to good use, and to find out what targets are worthy of designing drugs for, target identification, as well as basic science research. So in 1993, we published what turned out to be a seminal work, creation of a culture medium that did not have any serum components in it. It had only defined components. And we optimized that culture medium, as I'll show you in a minute. And that medium, NeuroBasal B27, has been kept as a trade secret all these years and has been useful to a number of scientists and now pharmaceutical companies as well. We extended that work in 1997 to be able to get neurons out of an adult brain. So neurons are all connected with synapses, and we were able to somewhat gently pull them apart, keep the cell body alive, and then get the cell body to regenerate with axons and dendrites. That particular direction resulted in a patent of a medium called hibernate that keeps the neurons alive outside the incubator. That enabled us to supply brain cells to anybody who would write to us and cared about having them. Usually rodent, rat, or mouse brain cells, and we could ship them overnight. So it required the conjunction of FedEx and UPS working with a neuroscientist to be able to ship brain cells all over the world. But we had trouble getting that license, that technology license, and after building the business a little bit in the university, getting in the way of the regular research activities, and I said, oh, I gotta form a company. I can't just let this die. So we started a company just a couple blocks away from the university, bought a building, bought some equipment, and within a year we had our debt fully paid off, and from ever since it's been positive cash flow. In 2008 we further advanced this medium, and I'll show you some evidence that we can create more synapses, more connections between the nerve cells than we were before by process optimization, and that these higher spike rates were advantageous then for better communication. This is the NB Active 4 medium. And we were able to start a branch in the United Kingdom as well. So here's the NeuroBasal B27 medium. It's really not tracking here. What happened? My computer is advancing, and I didn't even look at the screen. I'm going to try pulling the plug. neurobasal B27 medium contains a number of amino acids. There we go. In the middle there, a number of inorganic salts, which were pretty well understood as being biocompatible and bionecessary. But we added some antioxidants because normally your tissue, except for lung tissue, is not exposed to such high concentrations of oxygen. And some hormones, we optimized the dose response in terms of viability of the neurons. And as a result, the chart on the left shows that the density of neurons that could be supported in the dark circles at the bottom, we could increase quite dramatically in this optimized culture medium, the open circles. So each of these was did a dose response curve, some process optimization on each of these to produce a superior medium that is still made the same way as it was, 22 years ago. We thought, well, this worked well for embryonic neurons. That's all anybody did, and it's about 4 publications of taking adult neurons from either humans or from surgery or from an adult rodent and putting them in culture so you could do multiplicative experiments and determine dose response characteristics. This didn't respond again. I don't want to pull the plug every time. It was advancing, wasn't it? Perfect.
Has it moved on your laptop?
Yeah, the laptop is fine.
Exit presentation.
Let's try some kind of syncing. Not control this very well. It's like it's got a mind of its own.
It's the definition of intelligence.
Do you extending the screen or copying the screen?
It's now copying the screen. Try to look in this mode because now I can accomplish a one-to-one relationship. Here we can make this a little smaller, this a little bigger. And notes. So the problem with working with adult neurons, number one, you got to pull them apart from all the other neurons. And while you're doing this, you can't be inside of an incubator where neurons are normally cultured. So your body has this incubating system that controls the pH with bicarbonate as a buffer, a pH buffer. And so we adjusted the bicarbonate concentration and we're able to now work for hours and hours outside of an incubator, keeping the cells alive. And that's what led to the hibernate pattern. And this shows that we can get neurons and get them, this is 2 weeks in culture, and they're starting to connect to each other and form synapses.
What's the magnification of this?
Each cell body here is about 10 micrometers. So that's 3 centimeters. So that's 100 times 30. 3,000 magnification. That's not really that tremendous because all the action actually occurs right here, these things, these connections. And think I'll get to this in a few minutes, but your brain has one of those connections almost every micrometer, every 10 to the minus 6 meters, every 10 to the minus 3 millimeters. Very high connectivity. So we thought if we can grow neurons from an old animal, or an animal of any age, then we could study aging. And what's one of the biggest plagues in aging in the brain is Alzheimer's disease. So this is your brain on Alzheimer's disease, and this is a normal brain. You see some shrinkage over here. That's because there's a lot of synapse loss. And here's a measure of your cognitive ability once you get Alzheimer's disease, and usually it starts a couple years before you actually get a diagnosis, and then you have about a 10-year course of decline that's unrelenting, and we know nothing we can do to stop that still today.
How does that compare to just cognitive impairment due to aging? without
Aging,
disease?
normal aging, you might go from 30 down to 28.
At the end of your life.
Pardon me?
At the end of life.
No, I'd say for the average age of death, maybe due to pneumonia or heart disease, because my grandmother lived to be 103, and she seemed pretty cognitively intact until about 98. But some people will say everybody gets Alzheimer's disease if they live long enough, if they avoid cancer and heart disease and a bad infection. But here's the demographics that's really scary, that this is accelerating because of the baby boomer, and 23% of our population in a couple decades, 23% of the people over 65 will have Alzheimer's disease. The current trends continue. And at this point in 2040, there'll be more cases of Alzheimer's disease than cancer. It'll be the second leading cause of death. So this is indeed a public health crisis that needs attention. Our theory, working on this, is really a systems theory, because Alzheimer's and aging are both complex diseases. So we kind of loosely know what aging is, but what you may not realize until you get there is that how damaging a sedentary lifestyle is to the age, contributing to the accelerating the aging process, and your susceptibility to stress. So one of the definitions of aging is that you don't handle stress as well. There's the classic thing of that back east, the guy goes out with a snow shovel, once a year and starts shoveling snow and gets a heart attack, where he's been doing that all his life. And it's just the aging process interacting with the exertion of the exercise. So we hypothesize that these things contribute, but they contribute to a biochemical shift that we call a redox shift, that I'm not going to take the time today to explain, but if you're interested, I'll be glad to talk later with you about it. And then that changes the control of the DNA, epigenetic changes that contribute to aging, and this ends up being a downward spiral then. But these green arrows indicate hope. You're probably aware that exercise is good for you. I don't know how much listening to this talk will be good for you, but in general, cognitive activity is good for you. We've discovered some other things that can help in shifting this redox state and reversing some aspects of this the bioenergetics of the brain cells. Then there's also a building field of epigenetic modulators. And you're going to be hearing about things that people taking a pill for depression and it cures them in one day because it's an epigenetic modulator, and powerful anti-cancer drugs that are epigenetic modifiers. So this is my systems biology approach, and actually my PhD student, Yue Dong, is here, and she's going to find out how all this biochemical pathways fit together, and how we can use that information. Here's some of the evidence we found early on that neurons that survive just as well in culture are more susceptible to stressors. So here's what the neurons look like after 3 hours after they're isolated from the brain. They're just round cells like most cells in the body. But after 4 days, you can see the beginning of processes developing that are going to reach out and touch someone. And after 3 weeks, there's actually quite a bit of communication between the cells.
That's like regeneration.
It is.
I thought neural cells cannot regenerate.
Right, and that's because you read old textbooks. They do regenerate, and actually there are newborn neurons in people's brains at our age as well. And that's a hot new field.
So there's a hope for spinal cord injury repair?
There's hope for spinal cord repair, possibly with pluripotent stem cells, induced pluripotent stem cells that they may get from a cheek swab or a skin sample that they can change by epigenetic programming, change them into neurons or heart cells or pancreas cells if we got diabetes, and that's coming on the horizon.
Thank you.
Now, please, anybody interrupt if you have questions at any point. I should say that here's the 2 examples of susceptibility. We took neurons from 3 different age rats, either embryonic rats, middle-aged rats are about 1-year-old, old rats are 2-year-old, near the median lifespan of a rat. And this is the fractional killing in the presence of this protein A beta that accumulates in the brain of Alzheimer's disease patients. And you can see that increasing amounts of A beta, yes, they're toxic even to young neurons, but more toxic to middle age and even more toxic to old age. And the shape of the curve here is consistent with single hit kinetics. So this is not a real complex process. It's just A beta acting on one target to kill the neurons. Glutamate is an excitatory transmitter in your brain, the most prominent one. And that also has this age-related susceptibility. So we'd like to bend this curve down to make it more like middle age. We have some evidence that we can do that. Here's one piece of that evidence that we found that even male rats, if they were given estrogen, they could improve their calcium dynamics and their survival against those 2 toxic agents, glutamate or A beta. So here's the normal toxicity curve for A beta, but if we give these old neurons estrogen, a very low amount, so it's not a feminizing amount, 10 nanograms per mil,